Welcome to the lecture for molecular techniques, where we're just going to go over the basics, the background or fundamentals that you have to be aware of behind the various molecular techniques which are required not only to identify viral infections, the cause of viral infections, but also for certain bacteria. We've developed molecular techniques for rapid testing. So molecular is really the wave. That's where the laboratory is going in the future. Even today, a large percentage of the microbiology lab is moving towards molecular identification. And also they're moving towards identification of sensitivities using molecular techniques. That's not as widespread, and there's a limited the limitations really are the number of tests you'd have to do and to get a full sensitivity. So, but it is where all the laboratories are going, including microbiology. There are several reasons why we would think about molecular techniques for various identifications and procedures in microbiology. Key among them is precision, obviously. Precision or being able to precisely identify the organism using a molecular label, using a molecular tag, gives us really very close to 100% accuracy on our identifications. The models that we use now with our biochemical testing, we don't necessarily get that kind of security in our identification. Organisms do weird things, and so they may not do something we expect them to do, or vice versa, and that sometimes throws off the reliability of our identifications. So obviously with precision, you're always going to get the same result if you test with the same molecular tag. That 100% of the time you're going to get the same result. Accuracy, you're going to know that the result is correct. So between those two, molecular techniques provide an attractive alternative to biochemical testing and cell cultures and fluorescent antibodies and things like that. In addition, the turnaround time of molecular techniques, while they're not as fast as certain instruments in, say, the chemistry or the hematology lab, when you're talking about the timelines that microbiology uses for culturing, molecular techniques are far and away better. You're talking about turnaround times of a few hours, in some cases a few minutes, depending on what the technique is, from setup. In the case of routine testing, a culture takes a minimum of 24 hours, and we rarely return cultures in 24 hours. We usually like to keep them for 48 because of those slow growers. So the turnaround time is wildly improved with molecular testing. One of the problems, though, with molecular techniques really is the difficulty in performing. The training, the equipment, and the reagents all are impediments, really, to implementing molecular techniques. Now, while younger technologists would have more exposure to these molecular techniques during their training, us old folks, we didn't get a lot of this because it wasn't available at the educational level when we were being trained. That doesn't mean it's not possible to train any medical technologist in this, but the learning curve can be steeper, in particular for your older, less enthused by change technologists. The equipment is a large investment, and the reagents certainly are sensitive. Some reagents have to be made on a daily basis. So these are all things that have to be considered when you consider whether or not to employ molecular techniques in your laboratory. So the basics for molecular, this is just stuff you already remember from biology, from your biology class. But just to review them very quickly about your genome, basically. Remember that your genome, or the chromosomes in your body, contain all the genes that your body needs to function, and those are contained for the most part in the nucleus, although there are genes contained in the mitochondria. Now, when we're talking about viruses and bacteria, there's no nucleus and there's no mitochondria, but they still have genes. There still are chromosomal segments and genetic material. In bacteria, it is DNA. In viruses, it can be DNA or RNA. So that's important to remember. In addition, you can use, even in an organism that has DNA, you can use an RNA target still. Ribosomal RNA is a frequent target because we know the specific sequences are unique, obviously, to that organism, if they are unique to that organism. Now remember, not all of the genes that are present in any organism are necessarily expressed in every cell in an organism. In our cases, we're only talking about one cell. And that the molecule DNA in the chromosomal molecule is normally very highly coiled, which means that it's normally inaccessible to all of those enzymes that are needed for the process of transcription. Remember what transcription is. The production of an RNA sequence off of a DNA template. Okay, and so some of the enzymes that are required in order to do that are polymerase, some endonucleases, DNA ligase, exonuclease, helicase, and methyltransferase. These are all enzymes that should be completely familiar to you because you took biology 1 and 2, and so you know that those are required to uncoil that normally highly coiled DNA to allow the RNA molecules to pair up and to cut out damaged sequences, and then finally recoil the molecule behind it. There's a lot that has to go on, transferring nucleotides, connecting nucleotides, and all that stuff. Remember also that the nucleotide which attaches to the template is dependent upon the complementarity of the bases. So based on the sequence of your DNA, you're going to get a known sequence of DNA because we know that adenine binds with uracil and guanine binds with cytosine. Remember those, and then of course you have thymine, which binds with adenine. So those things are the rules that apply to DNA and RNA binding when you're undergoing transcription. Now, I know you learned in biology that this is a rule, but you also know that biology doesn't really like rules, and so mistakes can happen. And when they do happen, they have to be recognized by, pardon me, by molecules within the nucleus that will then cut out the damaged pieces that hopefully can be repaired. There is an editing feature in all RNA production, but the reality is we don't know if we're going to actually fix it properly. We don't know if our editor is really all that good at it. Then some other things to remember. These are just things I'm hoping to remind you of. You remember that the chromosome itself actually consists of sisters, right? The chromosome is 2 pairs. The chromatids are held together by a centromere. Remember that? So the paired chromosomes stay together. They hang out together, so that the transcription will occur to one or the other chromosome, and that depends, dominance versus recessive genes. If they're homozygous, it won't really matter. And then at the end of every chromatid, you have this highly repetitive sequence that sort of caps off the end of the chromatid. Remember that's called a telomere. And that telomere has protective functions on that chromatid, okay? As you age, the telomere gets shorter and shorter, and that's why you have less and less protection from those mistakes that can occur as you get older, unfortunately. For any cell, in order for transcription to occur in any cell, including bacterial, including viral, the genes have to be accessed. In order for that to happen, you need a a small sequence called a promoter to be able to bind to a particular area, or the area on the gene called the promoter to be bound in order to start the process of transcription. So you have a couple of things that have to happen. The chromosome has to be unwound in the correct place, and that will be determined by the various transcription factors that are released in the cell. So if a particular transcription factor is released, it favors a particular area of the chromosome. So they target certain genes, and then the promoter can be bound, and transcription can take place. Polymerase can start to add on nucleotides. In addition, there are several regulatory stretches in any gene which will control how and when a gene is going to be expressed. And they don't become part of the final sequence of the amino acids. They just control under the influence of other chemicals, other molecules that are present. They either increase the transcription of a certain gene or decrease the transcription of a certain gene. Okay, it's a feedback mechanism. Also remember that, as is shown in the little cartoon there, it takes 3 nucleotides to code for any single amino acid, and that'll occur at the ribosome during translation, which is really the goal for this whole process. Okay, so to go through these little players that I have listed here, you can see that the players in transcription are obviously the polymerases. There's several of them. And they're able to polymerize nucleotides, connect the nucleotides to each other. They basically they encourage or enable pyrophosphorylasis and pyrophosphate exchange between the nucleotides, so that you end up with a connection, a bound, right? You produce a chain. And there are various types of polymerases. If you've done any kind of PCR, you know that is true. Then you also need exonucleases, and these are all enzymes that you'll find you'll need during PCR. Many polymerases have an exonuclease attached to it. So a particular part of the polymerase has an exonuclease function, and what that does is it kind of has a proofreading capability. That's where it's kind of built in to the polymerase, so it notes that exonuclease, which the whole purpose of the exonuclease is to cut out nucleotides. So it notes if there is an error in the pairing, and it can cut out the last nucleotide. We have some terminal transferases, which is a type of a polymerase that can actually create a chain without a template, and that's thought to have a very significant role in telomere production. Then we have the endonucleases. Endonucleases are there to break the sugar phosphate backbone of the DNA. Okay, a particular class of endonucleases called restriction enzymes. These are various classes, and you would have different types of restriction enzymes that recognize specific base sequences in the DNA sequence and are able to break that sugar phosphate backbone at that juncture. We use endonucleases or restriction enzymes in PCR when we want to only copy a particular sequence of DNA, and we know what the beginning sequence is and we know what the end sequence is. So we use restriction enzymes to cut out that sequence of the DNA, the entire DNA length. Pardon me. A lot of times we can isolate these restriction enzymes from various different cell types, because these actually originate as part of the immune defense of any cell, since they've been evolved to recognize bacterial sequences. So we can actually extract these from your DNA, from your cells, from your cell types, and we can use them to target various bacterial targets, because our immune system has a normal host response against those sequences. And then we can also find them in for targeting viral sequences in other cell types, other animal types. Then we have DNA ligase. DNA ligase is able to form a phosphodiester bond between adjacent 3' hydroxyl and 5' phosphoryl nucleotide ends. So basically, this is the guy that's going to bring the two links together. Then we have an exonuclease. This is, I kind of already talked about this, but as a general rule, any exonuclease is an enzyme that degrades DNA, but it'll only degrade DNA from the free ends. You would need an endonuclease to cut it in the center. Also, if you have a circular DNA, since there is no free end, it can't do anything, okay? So that won't work against a bacterial genome. And there are lots of different exonucleases that have been isolated from a variety of different bugs. Bacteria produce a lot of exonucleases because phage genes, bacteriophage genes, which are viruses that attack bacteria, they're usually linear sequences, and the bacteria produces them to eat away at those sequences from the ends. Won't hurt their own because there is no end. Then we have helicases. Helicases control the organization of DNA, in particular that supercoil. And so we need helicases to be able to break down that supercoil or get the DNA out of that supercoil so that we can undergo our cutting, all the things that we need to do. There are two types of helicases, so you're aware of them. Topoisomerases, and those topoisomerases allow that super twisted DNA to relax, and then gyrases, which can actually untangle the DNA by producing double-stranded breaks so that it's not tangled. And then finally, we have methyltransferases. Sometimes we need to use those in our procedures, and all those do, which from the name you can probably tell, is they add a methyl group to the nitrogen bases, which is necessary, in particular for those adenine and cytosine nucleotides. In prokaryotic DNA, when that's our target, they usually use, most prokaryotic DNA is methylated only on one strand, and that's going to differentiate it from eukaryotic DNA, which is methylated only in specific regions. So we need to know where or what our target is and whether or not we're going to need this methyltransferase. Then a translation, I'm just going to remind you of a few key points. These are things you need to know because they're going to show up on a test, including your boards. And so I'm just reminding you of them very quickly because you already went through this in Bio 201 probably. So if you remember, during the process of initiation, the promoter is a key player. That's the site in the DNA sequence that signals the initiation point for a particular protein sequence. In other words, that's the site that the RNA polymerase will identify and will be able to assemble at that site. Once the RNA polymerase assembles, elongation can occur where an antisense strand is produced, meaning you're producing something that's going to be, a messenger RNA, okay? And remember that that's going to be opposite to the DNA sequence. So just remember that uracil replaces thymine in translation. And then termination, finally, it's a little bit different in eukaryotes than in prokaryotes. In eukaryotes, the RNA polymerase moves on until it reaches a sequence called the polyadenylation signal, or the poly A, which is just a bunch of adenines, one after the other. And that's actually the signal for the RNA polymerase to fall off, to disassemble. Most messenger RNAs will also have a poly A tail, but isn't produced here. That's produced in the editing, if you remember, of that messenger RNA sequence. So that's important for us. It's sort of an added feature in eukaryotes. And we have also, well, the enzyme that we use in those eukaryotes where we want that poly A is called the polyadenylate polymerase, if you remember, and that adds all the adenines to the end of the messenger RNA transcript. There's usually about 200 adenines per transcript added, so it protects it as it moves through the cytosol from being degraded. Because remember, it's dangerous out there for these molecules. These are just some basics. Make sure that you remind yourself about transcription and translation. Obviously, I didn't go through all of it. I just went through some of the key players. These are ones that have shown up and do show up on tests, so I want you to be aware of them. Then we have a little bit of a primer on nucleic acid. You can watch this video at your leisure. I'm not going to play it right now. It's just a YouTube video, which you should be able to find in online. Just to remind you that DNA or deoxyribonucleic acid is what our genome is as eukaryotes, and it consists of coding regions which are interrupted by long stretches of non-coding regions. And I want you to remember this because you probably always mixed it up when you were in Bio 102 or genetics. But the non-coding regions are called introns, which make you crazy, and the coding regions are called exons, just to make it crazy for you. And they're both going to be included in your initial RNA transcript. When you produce your transcript off of your DNA, it's actually a lot longer than it needs to be, and so it has to be edited. And so you get the initial product is called HN RNA, or heteronuclear RNA, and that will be edited in the nucleus to remove all of those introns. And those that'll be done by the process of splicing, where you remove those intervening sequences and and hook up all the exons together. It is possible that you can have one HN RNA which can be spliced differently to produce multiple mature mRNAs simply by using different combinations of exons. Okay, and that will depend on the surrounding environmental conditions in your nucleus, which will be determined by transcription factors and all those other chemicals, but you can end up with different products in that manner. Then the messenger, and same way you get different RNA, ribosomal RNAs and transfer RNAs. So that means that each one of these steps, each one of these products is basically another potential target for us to use where we can look for unique sequences or products that allow us to identify various organisms. And we can target these in our own cells, so maybe cells that are easy to get to, like for example, well, you want a cell with a nucleus, so red cells don't have a nucleus. They're not something that we would be interested in. But white cells are pretty ideal because they're in the peripheral blood, and not only that, they have a lobulated nucleus. A lobulated nucleus because they actually have multiple copies of the entire genome, if you remember correctly. Okay, the multiple copies, that allows us lots more targets. Okay. In addition, we can look for them in viruses and bacteria by growing up the population. Again, multiple targets. We're increasing exponentially the targets that are there that we can look for. But we have to remember the nature of the target that we're looking at. Is it DNA? Is it RNA? Is it messenger, ribosomal, or transfer RNA? Most of our targets will require some kind of pretreatment based on their source or whatever the target nature is, so that we can amplify our chances of actually finding that little thing that we're looking for, the needle in the haystack. So for example, if you're looking for one particular section of a gene, if your target is a sequence of one particular section of a gene, there's a lot of things you need to do. You need to get the DNA out of the nucleus where it's protected. You need to separate it from everything else in the cell. You need to unwind it. You need to open it up and release those nucleotides so that you can find the particular sequence that you're looking for. Okay? And the next, then when we're talking about nucleic acid extraction, remember that our primary purpose is to release the nucleic acid from the cell, whatever our target is, so that we can use it for the next step. There's a lot of processes here that sound like they're the whole test. But like when a doctor orders PCR, that's kind of limited. He doesn't want you to just replicate a bunch of genes. He wants you to do something with that afterwards. So PCR is actually a misnomer for what we're going to be doing. PCR is only a part of what we're going to be doing in this entire process. And all of it needs to start with nucleic acid extraction. And the purpose is to release that nucleic acid so that we can use it for the next step, whatever it's going to be. We want to release it so that we can get the nucleic acid completely uncontaminated. We want to remove it from all the other proteins, carbs, and lipids, and even the other nucleic acids that we're not interested in. We really want either pure RNA or pure DNA in our test tube so that that's what we can test. Why should we have to weed through all of the hay? Why can't we just separate out all the needles that are in there? So this is going to require some significant pretreatment, you can see here. Initially, it's going to be done by lysing the cell. Okay, we got to get rid of all those membranes, including the cell and the nuclear membrane, and you want to do that under conditions where you won't damage the actual nucleic acid. Then you need to purify the product and determine the concentration and the purity of the product before moving on to the next technique. So if we use our example of a white blood cell, because that is really one of the easiest cells to get at, it's just a little bit of phlebotomy and we get at it. So let's separate out the white cells out of the peripheral blood or the bone marrow, whichever you're using, by using differential density or gradient centrifugation or differential lysis. In hematology, you learned that red blood cells and white blood cells don't have the same kind of osmotic fragility in when you test them. Red cells are very easy to lyse. A little bit of water, bang, they burst. White cells will last a little bit longer. So if you use, well, water will lyse white cells, so don't use that. But if you use certain osmotic tensions in your fluids, you could lyse the red blood cells much easier and quicker than you would lyse any white cells that are in there. Okay. In a differential density gradient centrifugation, you basically mix either your blood or your bone marrow, whichever you're using, with isotonic saline, and then you overlay it with a solution like Ficol solution, or Ficol's reagent actually, which is actually a highly branched sucrose polymer that isn't able to penetrate biological membranes. And that'll allow you to separate and significantly enough the white blood cells from the rest of the cellular matter. Basically, your white blood cells are going to settle into a layer in the Ficol, which will be below the plasma components, which are less dense, but above the other cells, which are more dense. And then you just have to remove that Ficol layer. Normally, if you just do a buffy coat where you highly spin the cells, first of all, you can damage the cells, but your white blood cell layer can be very, very thin. So you add that phycol layer so that basically you're thickening that white blood cell layer. Now you're not necessarily interested in getting every single last white blood cell, but you want just white blood cells. And so that's where this procedure will allow you to remove those white cells with the phycol. Then you just have to wash off the phycol, right? Just wash it off a couple of times, and that'll allow you to get the white cells out. Now you've got pure white cells, so you can burst those membranes and remove and isolate the nucleic acids. Another process is differential lysis, where you have that difference I talked about in the osmotic fragility between the red and white cells. And what you do in that case, if you wanted to do that, is you use hypotonic buffers, including water. Water would work as well, which will lyse the red blood cells before the white blood cells. Then you gather the white blood cells. Now you don't have any other cell but the white blood cells left, and you just gather them into a button, leaving your lysed red cell components in the supernatant, and you can just pour those off. Once you've done that, now you have purified your cells, and you'll have to go through the next step in order to complete the testing. So this is one part of the procedure, a fairly extensive part of the procedure. One of the other things that you're going to have to do is you don't necessarily want to test the entire genome, okay? Or if it's proteins, you don't want to test every single protein. So one of the methods, one of the things that you're going to do is resolve, which means be able to focus on, right? Resolve all of the materials that you have or separate them according to their similarities, either by size or charge. And one of the methods that we do this is electrophoresis, okay? Now you can use electrophoresis to also resolve or separate nucleic acids. And now DNA or nucleic acid is negatively charged. We know that. Under an electric current, DNA is going to migrate towards the positive pole and negative towards positive. Okay, and the anode. So that we know. Wonderful. You can do electrophoresis either in a solution, but when you do it in a semi-solid or solid material like an agarose or a polyacrylamide gel, the migration is going to be limited based on the size of the DNA, and that will be based on the spaces in the cell. And so I give you this chart here, which shows you what concentration of agarose you would use to separate certain sequences of DNA. If they're very small, you can use a very highly concentrated or a higher concentration of say a polyacrylamide gel, or a higher concentration of agarose. If it's very low, you would use a lower concentration. Okay. Now each nucleotide has the equivalent of one negative charge, and so your charge to mass ratio of the various sized molecules will remain constant. So that means that the DNA fragments will migrate solely based on their size, and their charge won't come into it. Okay, since the charge won't change as the size increases. So this allows us to separate the DNA sequences based on size. Now we would separate them following maybe treatment with some kind of restriction enzyme that allows us to cut that DNA into the sizes that we're interested in. If we know that the gene we're looking for is 1,000 base pairs long and we know the sequences that it'll start with, well, let's cut first with a restriction endonuclease, and then if we know it's 1,000 base pairs long, we'll use an agarose at 0.6% gel, a 0.6% agarose gel, and we'll separate it out, and we'll be able to have only those sequences that we work with, everything else that we washed out. It's easier. We're shrinking the size of our haystack. Okay. And then the two things I just have down here, the agarose and the polyacrylamide gel, those allow us to really fine tune the kinds of the sizes of the sequences that we want. And you can see agarose you would use for much larger sequences. When you're talking about really, really small sequences, you would use a polyacrylamide. It is not without precedent to first separate in an agarose and then separate again in a polyacrylamide, right? You'll get much more fine tuning because you can see the ranges in the agarose are much larger than the ranges in the polyacrylamide. Just for some other types of electrophoresis, pulse field gel electrophoresis is a little bit different. We use this to isolate very large pieces of DNA, fragments of DNA between 50,000 and 250,000 base pairs. These wouldn't be easily resolved with simple agarose electrophoresis. Even in the lowest concentrations of agarose. So these are very large megabase fragments. And so we use a different method, okay? So you have to know that there are different methods out there available based on what's the sequence, how big is the sequence that we're looking for. But when we do that, basically what you're doing is you're having the charge go in different directions across the gel. Well, it says pulsing across the gel itself. Okay. There are several methods that are shown, and you can see the figure here that shows you the different methods for pulse field. The methods are based on different configurations, and you can see the switching of the current in the different directions. Whether it's just at an angle, at a 90-degree angle, or at really different, it's the angle between that separates them. Basically what this does, using the different types of pulse field, allows you to, because DNA doesn't really like to be in a charge, so it has a tendency to break. The larger the DNA, the higher the higher the probability of breakage. And so pulse field will allow allow you to protect your sequence that you're trying to resolve from breakage. Okay, and it'll be immobilized at a particular spot that you can predict based on size. Okay, but then once you have it immobilized, you're going to have further treatment that has to be, remember, you're going to have a piece of DNA immobilized in this agarose plug before you are even close to PCR, okay, before you've even gotten to there. So it'll probably have to be further edited or further treated with some kind of maybe a restriction enzyme or you can even do that while it's still in the agarose plug. There's no restrictions against that. You can immerse the agarose into a restriction endonuclease. And then you can insert that plug directly into another gel for routine electrophoresis. You don't have to necessarily isolate it from the agarose or anything like that. This is a common procedure that's used for full chromosome-sized fragments of DNA that we might do, that we might use, like for example, in bacterial fingerprinting, if we're looking for, or bacterial genotyping is another name for it. When we're looking to see if a particular bacteria is the cause of an outbreak. Okay, we can do that. In this case, we would need to use very, very long sequences of DNA. In hybridization technologies, you're aware of several of these. Just know that they're hybridization. These are methods where we can hybridize, where we use a probe that is antisense to our target. So you're producing, and it's usually a hybrid that we're producing. So if you have a DNA target, you use maybe an RNA probe so that you know that exactly what sequence that you can target. Now, it is possible to use a DNA probe against a DNA target. These hybridization techniques, you can, depending on the target, you know what kind of probe you can use. If it's a protein target, you can use an RNA probe. You can also use for these hybridization techniques. These techniques allow us to identify specific sequences that are present after we've resolved a particular section of DNA. So that means that, we resolve something on in an agar, and then, okay, so we've separated things. How do we know what's there? How do we know if the sequence that we're looking for is there? Well, we use a probe, and those probes are based on hybridization technologies. We know exactly what the sequence is. We can produce a probe that is antisense to that sequence, and then we overlay our gel or our nitrocellulose paper with that probe, and it will, usually those probes are tagged with some kind of enzyme or some kind of signal, so that we can then cut out the section that has our sequence of interest in it. The very beginning of hybridization technologies was with the Southern blot, named for Edwin Southern, who was the first to actually report the procedure. In the Southern blot, DNA is isolated and cut with restriction enzymes, then they're separated by gel electrophoresis, depurinated and denatured, but then they're transferred to a solid support because agar doesn't really stand up to time, but nitrocellulose does, so it's transferred to nitrocellulose. And then in the final steps of the procedure, this is where the hybridization comes in, the DNA fragments are exposed to some labeled probe of a specific sequence that we know. It would be complementary to the target sequence. And so if that target sequence is there, you'll have that bound probe in the regions of interest. Any unbound probe can be removed, washed off, because it's not bound to anything, and then the signal of the probe can be detected to indicate the presence or absence of the particular sequence that you're looking for. That's kind of what the whole procedure is written here. So you have the restriction enzymes, which will produce those fragments that you're interested in. You're going to denature the DNA so that it's open and separated from each other. You're going to separate it by electrophoresis and then blot it. And the blotting is shown here at the bottom in this little cartoon at the bottom, where you can use capillary transfer, electrophoretic transfer, vacuum transfer. You guys have already heard of all of these. And that allows you to have that sequence permanently bound to a membrane, which just means you can do more with it. Now, this is Southern blot. Since the evolution of Southern blot, there's several other type of blotting techniques, which just are based off of his name, Southern. Okay, it has nothing to do with being, having to be pointing north or being done in the north or anything like that. In Southern blot, your target is DNA. In Northern blot, your target is RNA. In Western blot, your target is protein. Okay. Now, the probes in Western blot are usually protein probes. They're usually antibody probes. You can also do a Southwestern blot, which is a mixture where you have a protein target with a nucleic acid probe, a DNA probe that would be complementary to that. Okay, the process, though, are very similar. It's just that your target is different. It's going to sound very similar. These are just cartoons of the two different types of transfer. Instead of just capillary transfer, you can see that we can enhance transfer. In capillary transfer, you kind of have to wait overnight for all of the molecules to move, to seep through all of the paper. So we're very lazy and we don't want to wait overnight. So, or I shouldn't say lazy, we're impatient. And so we have electrophoretic transfer. You add a charge, increase the rate of flow. Okay, that's something that we can do. It's not as cheap as capillary transfer, but it's faster. And then if we really want to get fancy and we have something that's probably even more expensive, we can do vacuum transfer, where we can apply a vacuum to the chamber and force the molecules through from the agar into the nitrocellulose. Then we have yet another technique to add to this process, which can be done if you've transferred something to nitrocellulose, or if you have something still, you can actually do it on a gel as well. It's easier if you transfer something to nitrocellulose. Now, if you know that you're going to use a hybridization technique at the end of separating out your molecules, if you're not going to use a blotting, well, you can still use a blotting technique. This can be post-blot, essentially. This requires, if you're going to use this, it requires a pre-hybridization step, okay? And in this step, what you do is you prepare the fragments that you separated for the binding of your probe. If you skip this step, you're going to get a whole lot of non-specific binding to a bunch of random sequences, which will cause basically kind of like a high background to your results. So the pre-hybridization step is where you incubate the membrane that you have, that you're working with, in the same buffer that you're eventually going to have the probe in. That allows you to prepare all of the sequences that are there for interaction with the probe that you're going to use with whatever the probe is. And it can take several hours, so it does add time to your procedure. Now remember, northern blot is for RNA, don't forget that. So in denaturing RNA, remember that actually RNA is actually denatured by the process of electrophoresis. The RNA has to be separated from the DNA to make sure that you get pure fragments. So RNA kind of likes to hang out with DNA if it's available. So you must be careful to make sure that you remove the DNA from the procedure, but leave the RNA alone. And one of the things that can happen, it has happened in labs, is the technologist is in using an environment where DNA was being denatured, whether it was in the electrophoretic probe or whether it was in the chamber or whatever. And so you have to be careful. And in a DNA electrophoresis, we're very commonly using RNase to eliminate any free RNA that might get in the way. Well, if you do that when you're doing northern blot, there goes your whole target. Okay, RNase works very well, and it'll ruin your results. So in an RNA environment, one of the things that we frequently use is formaldehyde. It works very clear to denature the DNA, leave the RNA, and still leave the RNA. However, formaldehyde does have to be removed before blotting because it'll interfere with the blotting procedure. So you just have to add an extra step where you wash it in water, just deionized water. And the reason you have to do that is the formaldehyde, while it won't denature the RNA, it'll prevent the RNA from binding to the nitrocellulose membrane. So we want to make sure that we follow every step, right? Follow every step, no shortcuts. Okay, the technologists always want to look for shortcuts. There's no shortcuts here. So those are two little things that you have to make sure you keep in mind for northern blot modifications in hybridization. In western blot, there's also some modifications that exist. Western blot is a very common technique in the in virology and immunology labs. And that's because we use it for analyzing protein. And the sample we frequently get in western blot is serum. That's where all our body protein, that's where all our blood proteins are anyway. So if you get serum, you don't have to really worry about cells. You can destroy any cells that are there. We're not worried about them. So you get rid of the cells and you extract in the serum. You can do, they usually use polyacrylamide gels for proteins, which allows you to separate the proteins according to molecular weight. You can also use IEF gels or isoelectric focusing gels. That'll separate them according to charge. Either one works very well. But remember, when you do run a Western blot, you have to run pre-stained molecular weights as standards, or these molecular weight standards with the samples that allow you to control for your charge, make sure you actually separated everything. And it also gives you a reference for the size of the product that you're ending up with. And those are commercially available. You just buy the commercially available standards that go along with the sequence of the protein, the size of the protein that you're going to use. Now, remember that what proteins go through a denaturing as well during the purification process, and remember that denaturing can actually affect the folding of the protein, which can then mean that it will affect the epitopes that are present that would then bond with whatever your probe is. Okay. Now, if that in order to accommodate the fact that denaturing does occur during our purification process, we have to make sure that we include in any Western blot modification a renaturing step where the protein can reform in the proper sense. The key thing is pH. Watch your pH. If your pH is off, your protein's not going to form properly, and then your epitopes are going to be affected. So you might get your protein separated, but you won't be able to have anything available for the binding of our probe. And again, you're going to use the same thing. You can use a capillary transfer to the nitrocellulose or to electrophoretic, whatever transfer mechanism that you want. And this cartoon gives you the steps that go through. You can read through those to be sure that you understand them as you go through the separation of those molecules. Now the probes. How do we decide on how do they get the probes? This is actually the limiting factor. We know we can separate the targets. We know we can get those out. The limiting factor is, well, there's two limiting factors. What is the probe? Which sequence is unique to this organism or virus, to this protein? Which sequence is the one that's very unique just to this one, so that we can be very, very sensitive? We have probes out there that can identify a group of organisms, not necessarily one organism. Okay, so that's important. And they can be made, these probes, we have DNA probes, RNA probes, protein probes. They can be made in a variety of ways. One of the common ones is you can clone the sequence in a plasmid and then isolate that sequence using restriction enzymes, which would then be purified by gel electrophoresis. You can isolate these probes from viral genomes, and we can also use in vitro organic synthesis of a particular sequence. That's only good for really short oligonucleotides because if it's really long, it's not practical to do it in an in vitro method. If you produce a DNA probe, those actually are pretty stable. You can create those probes and they can be stored for some time in your laboratory. Many of these laboratories have to produce their own, or they do produce their own. As you know, some laboratories have in-house tests, so the probes can be produced on their own. The length of the probe will determine the specificity of the probe. Okay, the longer the probe is, the more specific it will be. Okay, because any short sequence could occur randomly in anything. Okay. RNA probes are obviously going to be made from a DNA template. And because RNA and DNA tend to form a stronger helix than DNA, the RNA probes generally offer more sensitivity than DNA probes in the southern blot. So you actually produce the probe in its hybrid form, where you have the RNA, DNA, and then you include that denaturing step in your process to remove the DNA and free the probe, as it were. Okay. But you have to be sure that you have the correct complementary sequence for the target, since you won't have the luxury of two targets like you might have with a DNA probe. Okay. Your RNA probe, once you produce it, it's not as stable. And so it's going to have to be used within a few days of production. And of course, you have to add that step where you remove that DNA template from the probe. As long as you have an RNase-free DNase, you can do that pretty easily. And then we have a couple of specialized probes. We have a probe called a peptide nucleic acid probe and a locked nucleic acid probe. These are synthesized using special chemical methods, and what they have is modified nucleic acids, which actually provide the advantage of being resistant to certain nucleases that would degrade DNA and RNA. Those nucleases are normally present in your cellular material. So these are specialized probes that are produced to protect the probe from specimen material that might be present in the test environment. And then we have protein probes. Protein probes are almost always antibodies. As I said, there is a Southwestern blotting process where you can use a DNA probe to bind, but almost all, that's usually for research. In the clinical lab, almost all protein probes are antibodies. And we have two types. It could be polyclonal, which is pretty easily produced. You can immunize, say, an organism against a specific antigen, and then just collect the antibodies produced against that antigen in the organism. But if it's polyclonal, it's going to be reacting against various epitopes on the antigen. Okay. And unfortunately, with polyclonal, those various epitopes could also be found. Some of those various epitopes can also be found on other antigens present. Okay. A monoclonal antibody responds only to a single epitope on the antigen. And obviously, because of that, they would be more difficult to produce. It has to be purified. And basically, one of the mechanisms of doing this is through hybridomas produced when you fuse immunized spleen cells with mouse myeloma cells that'll produce a hybridoma. And that hybridoma will grow in culture and secrete antibodies. That's generally the production method of choice for monoclonal antibodies. As you can imagine, when you're purchasing things for your laboratory, polyclonal antibodies will be less expensive than monoclonal antibodies. So you have to weigh how specific do you need to be for this particular identification. Okay, so how do we detect? What probes are we going to attach to, or I should say, what detection methods are we going to attach to our probes so that we can see that the probe is attached to something? Okay. Well, originally, in the method developed by Dr. Southern, he used radioactivity, because everybody used radioactivity then. He used phosphate 32, which is very easily detected. So you allow the phosphate 32 reactive probe or labeled probe to interact with your nitrocellulose membrane. Allow that to happen for a specified amount of time, then wash off the unbound probe. And in the case of phosphate 32, you could use a light sensitive film to detect the hybridized and tagged probes. Okay. However, as you know, I've said time and time again, radioactive probes not attractive anymore in the laboratory because we don't like to have to dispose of them. Of course, there's a risk to your staff as well, but they're very expensive to get rid of. So there's a lot of non-radioactive probes. which can be bound covalently to either to several other markers such as deoxygenin or biotin. And then after those are hybridized, you can wash away the unbound probe and then use something like the anti-deoxygenin antibody, which will react with your deoxygenin, or streptavidin, which will react with your biotin. And those can be conjugated to an alkaline phosphatase that's added in the reaction mix to bind to either one of those labeled probes, to those labeled probe target complexes. And those allow you to detect them, okay? So in the case of light production, if it's hydrogen, the most common one used, I think you guys have been most exposed to horseradish peroxidase, and that can be used in this procedure as well. And then your detection method will depend on what your signal is. Is it light? In the case of the 32, is it radioactivity? Is it a chemogenic signal? You know, light is the most common method at this point. And so you use a luminometer to detect the light. Very simple. There are other detection possibilities, and you can see here horseradish peroxidase, HRP, which is our most common one, alkaline phosphatase. You guys have worked with both of these in the laboratory. Several reagents can work with those. So if these are tagged to your, or these are attached to your probe, to your tag in some way, and you add a conjugate or a reagent to them, you'll get different color precipitates, which you can visually see. In some cases, you can also use a colorimeter or a luminometer to detect the intensity of those precipitates, which will give you a semi-quantitative or quantitative measure. In a chemiluminescent situation, you can use the same enzymes, but add a chemiluminescent reagent, and that'll emit light, which can be read by again, a luminometer, and it can measure the intensity of the light. So here is another video which you can watch at your leisure. This is a DNA isolation technique, which just look through the tutorial. I'm not going to read it because it'll be read to you. And this is just for the extraction of nucleic acid. This is kind of just to go back and show you a cartoon of how it's done. For the PCR itself, which we're finally at now, so how do we perform PCR? The things that you need to remember are the three basic steps, or the three elements of any PCR cycle. And those elements are going to be separated from each other pretty easily, actually, by initially temperature. Temperature is one of the most important differentiating things in these between these steps, and then of course time. So the first step in any PCR procedure is denaturing, where the double-stranded DNA, we're going to use DNA, is denatured into two single strands in order to be replicated. The next, and it's the most critical step for the specificity of the PCR, is the annealing step. And this is the second step of PCR, and you can see its temperature is completely different, 50 to 70 versus the 90 degrees of denaturing. And in the second step, you're going to have two oligonucleotides that are going to prime the synthesis of DNA and anneal or hybridize to complementary sequences on the template. This requires that you have the primers. You have to have the primers for the sequence that you're interested in. And those primers will dictate which part of the DNA will be amplified. You don't have to amplify the entire template DNA. It's just the gene, the one gene that you're interested in, okay? So it's the primers that will determine the specificity of the amplification, sorry. Then the third and the last step of PCR cycle is the primer extension step. And this is essentially where the DNA synthesis occurs. So your replication is occurring here. This is where you're going to add your polymerase. Okay, the polymerase is going to synthesize a copy of the DNA of the template DNA by adding the nucleotides to the hybridized primers. Okay, the DNA polymerase will catalyze the formation of basically a phosphodiester bond between any incoming nucleotide molecules, which you also have to add to the environment, and that'll be determined by the hydrogen bonding to the template. Okay, A to T, C to G. Okay, you know that. And the DNA polymerase is going to add one base after the other to that 3' end of the primer. Okay, so the things that you have to have are a specific primer for the sequence you want copied, the polymerase that you need to bind the nucleotides to that primer, and then you have to add nucleotides. Can't forget nucleotides. If you and this whole thing is happening in your reaction vessel, you're changing the temperatures, and it takes a different amount of time. At the end of one cycle of PCR, you are going to have doubled the number of double-stranded DNAs that you started with in that first step. And you can see in the cartoon that I have here, you haven't replicated the entire sequence. You've only replicated the target sequence. Okay, so then in the next cycle, you're going to start cycle number 2 now. In the next cycle, you are going to denature those 2 pairs, and you're going to anneal the primers. You're going to have to add more primers because now you're going to have double the amount of targets. You're going to have more primer. Usually you should have enough primer there for the whole thing. So you're going to add the primer again and nucleotides so that the whole process can occur over and over again. And you can see at the bottom here, you start at, if you were to start out with a single copy, and this with the presumption that you started off with a single copy, at the end of your 35th cycle, you will have 68 billion copies, which is something you can detect now. Okay, that's something we can really work with in the laboratory. Okay, so be aware it's an exponential increase, so you should be able to do the math. Okay, if it's 2 to the 4, that's 32 copies. Okay, so you go up exponentially. Then this is just another a couple of charts, a couple of pictures for you. Hopefully it help you identify the importance of the primer. And you can see that the primer will have to bind to the areas just outside the area where you're interested, that target that you're looking for. Okay. And then here are some reagent components that you're going to need for the typical PCR reaction. Okay, and you can read through those. Everything, the primers, the nucleotides, the buffers, and the molecules that you need to create the right environment for the process to occur. Now, by far, contamination is the bane of all PCR methods, and contamination is going to be controlled both physically and chemically in your PCR lab. Physically, the best way to avoid PCR carryover is to separate the pre-PCR areas from the post-PCR analysis areas. You need to have positive airflow, airlocks. So in other words, engineering measures have to be taken, extensive measures have to be taken, in particular in high throughput laboratories that process a large number of these samples. And if you're testing for a limited number of amplification targets, all of these things have to be physically and chemically controlled so that you don't PCR, that the DNA can get everywhere. It'll cling on you, so you never go backwards. You never walk backwards through the door to the PCR areas. Equipment, including you, the laboratory, you're wearing your laboratory gown, your gloves, all that stuff, all those reagents have to be dedicated to either the pre or the post PCR areas. Items can flow from the pre to the post, but they can never go backwards, never go in the opposite direction without decontaminating first. Now, one of our major sources of decontamination is UV light. UV light has been used and is used to decontaminate and maintain those pre-PCR areas. And that's because the UV light, as you know, will catalyze single and double strand breaks in the DNA, which would interfere with replication. So you end up with DNA still there, but its ability to be replicated is removed. So all isolation cabinets, all PCR cabinets and equipment are equipped with UV light sources that should be turned on for about 20 minutes after the box has been used. And that allows any residual DNA to be damaged so that it cannot contaminate your next run. Now you can increase the effectiveness of the UV light by adding some chemicals like psoralins to the amplification process products after analysis. And those psoralins will actually intercalate between the bases of double-stranded DNA in the presence of long-way UV light. They basically covalently attach to the thiamines and the uracils and the DNA chain, DNA RNA chains. And what they do, these psoralins, they're really bulky additives, and they prevent denaturation and amplification of the treated DNA. So you don't want to do this for your target DNA. You do this for the products that are left over. The other thing, of course, a little bit cheaper, is decontamination of the workspace with a 10% bleach solution. So frequently wiping down your bench tops, your hoods, any surface that comes in contact with any specimen material with dilute bleach or alcohol will remove most DNA contamination. There's several other methods, commercial products that are also available in the thing. Those are the basic ones that help in most laboratories. PCR modifications that are present. These are just a couple of them, just so that you recognize the terms, okay? We have, of course, reverse transcriptase RNA, which is the amplification of PCR where since PCR requires a double-stranded DNA template, if the starting material is RNA, you can actually convert it into a double-stranded DNA using RNA, reverse transcriptase. So it's just the adding of that step for a specimen where you know that the beginning product is RNA. In nested PCR, this is a modification that increases the sensitivity and specificity of the reaction. Basically in nested PCR, what you use two pairs of primers to amplify a single target in two separate PCR runs. The second pair of primers are designed to bind slightly inside the binding site of the first pair of primers, and that'll allow amplification of the product of the first PCR in a second round of amplification. In the process of real-time PCR, which is a big thing, doctors love that term. I want real-time PCR, okay? And that's because this technique is based on PCR, the regular PCR reaction. It uses that, but it uses computers and the current technology that we have to monitor the amplification of the targeted DNA during the process, so after each sequence. And in real time, that's what they mean by real-timed, not only as an end product, okay? Like in conventional PCR. In conventional PCR, we just want to know, is it there or is it not? In real-time PCR, we can actually watch that exponential increase of the target, which allows us to do the math and produce either quantitative or semi-quantitative results so that we know how much of the target was in the original specimen. That's the idea. Okay, so these are where routine PCR is usually reported qualitatively. It's either positive or negative. Real-time PCR gives you a semi-quantitative or quantitative result, which and this is the one that's used for a lot of our viral loads. So if a doctor wants to know not just does the patient have HIV, but how much HIV are we talking about? What's the viral load? The implication would be that the higher the viral load, the worse the clinical symptoms the patient would be experiencing. That doesn't necessarily always ring true, but it is something that doctors want to know. And the other thing they can use it for example, in Hep C, they use it to be able to determine, because remember in hepatitis C, we actually have the word cure being used with some of these treatments. So if you do a real-time PCR and you find out there's none in the patient's sample, that's how we come out with that, that we can come out with that result.